friends, today's topic is protein synthesis or gene expression. Protein synthesis occurs in two steps, transcription and translation. Synthesis of mRNA from DNA is known as transcription, whereas synthesis of protein from mRNA is known as translation. Synthesis of protein from mRNA occurs on ribosomes, and the synthesis of protein in two steps, that is transcription and translation. They are collectively known as central dogma. Protein synthesis occurs in two steps, as I told you, that is transcription and translation. First we take transcription. Transcription, as I told you, the synthesis of mRNA from DNA is known as transcription. It also has three steps or three phases. In first phase, that is initiation phase, there is attachment of RNA polymerase enzyme to the DNA at its promoter site. And for this, the sigma factor of the RNA polymerase helps in attachment of RNA polymerase to the promoter sites. There are various promoter sites in prokaryotes and eukaryotes. In prokaryotes, these are at -10 and -35, whereas in eukaryotes, these are at -25 and -70. -10 is known as Tata egg, -35 is known as TTGACA, -25 is known as Tata, -75 is known as cat. So at the end of the initiation phase, there is formation of transcription bubble due to attachment of RNA with the DNA. So in transcription bubble, there are two different strands, sense and antisense. Antisense strand is also known as template. Now second step in transcription is elongation. In elongation, there is synthesis of mRNA in the transcription bubble. And in synthesis of mRNA, there is the addition of nucleotide in the complementary manner to the nucleotides present in DNA. mRNA synthesis always occur in 5 to 3 direction. Third is termination steps. In termination step, there is termination of mRNA synthesis and it gets separated from the DNA. When there is appearance of GC base pairs followed by AT base pairs in the DNA, then mRNA makes GC hairpins. It gets turned at this point and it gets separated from the DNA. After transcription, there is some modification in separated mRNA. It is required because there are various enzymes in the cytoplasm which might destroy mRNA. So there are two modifications in mRNA. First one is addition of 7-methyl GTP cap at 5 end of the mRNA and poly A tail at 3 end of the mRNA. And second modification is removal of intron. This is also known as splicing. And intron is non-coding part of the mRNA, whereas exons are the coding part. That means these have message to synthesize protein. So it was all about the transcription. Now we understand translation. Translation, as I told you, translation is synthesis of protein from mRNA. It also occurs in 4 steps. First one is amino acid activation. In this, various amino acids are activated by ATP and they form AMP, activated amino acid. Then they bind to the tRNA, particularly at 3 end of the tRNA and form amino acid tRNA as shown in this figure. Second step is that there is formation of initiation complex. It requires 3 things: amino acid tRNA, which was formed in last step. Second is smaller unit of ribosomes, and third one is 5' of mRNA. These three form the initiation complex as shown in this figure. The smaller green part is smaller unit of ribosome, larger part is larger unit of ribosome. There is mRNA 5 to 3' and there is tRNA with activated amino acid. In tRNA there are anticodon, that means the nucleotide which are complementary to the mRNA. If in starting there is start codon present in mRNA, that is AUG, as we know, there will be a anticodon in tRNA RUAC, and first amino acid will be methionine always. Third step in translation is elongation step. In elongation step, there are attachment of a number of amino acid for protein synthesis. There are 3 sites in ribosome for this: A site, P site and E site. At E site, there is exit of tRNA after giving amino acid, and A sites is for addition of new amino acid with tRNA, and P sites forms peptide bond. In second diagram, we can see there is an enzyme, that is peptidyl transferase, which transfer first amino acid, that is methionine, to the next amino acid tRNA complex. So there is formation of or addition of various amino acids on ribosomes. Last step is termination. Termination, at the starting of termination, there is a long chain of amino acid on ribosomes. But at termination, there is appearance of a stop codon on the mRNA, so there will be no synthesis or no addition of more amino acid on the ribosome. So no more protein synthesis. So a factor, that is releasing factor, helps in termination. In protein synthesis, there is movement of only ribosomes, not the mRNA. So as the ribosome moves and as there is appearance of stop codon on mRNA, there will be stoppage of protein synthesis. So this was all about the protein synthesis.